Hello viewers, welcome to the course Elements of Genetics, and in today's part we shall be studying about nucleic acid as genetic material, replication, genetic code and translation. About the nucleic acids, we can say that it was Friedrich Mischer in 1869 who isolated what he called nuclein from the nuclei of the pus cells. Nuclein was shown to have acidic properties, hence it became called nucleic acid. There were two types of nucleic acids were found, deoxyribose nucleic acid, which we popularly called as DNA, and ribonucleic acid, which we call RNA. The distribution of nucleic acid in the eukaryotic cell, as we know that there are two kinds of cell, the prokaryotic and the eukaryotic cell. The DNA is found in the nucleus with a small amount in mitochondria and chloroplast. RNA is found throughout the cell. This is very important to understand that DNA is also present in case of chloroplast and mitochondria besides the nucleus. DNA as genetic material, the circumstantial evidences were gathered from these evidences. Number one, that it is present in all cell and virtually restricted to the nucleus. Second, the amount of DNA in the somatic cell, that is body cell, of any given species is constant, like the number of chromosomes, and the DNA content of gamete, that is sex cell, is half that of the somatic cell, and in case of polyploidy, there is multiple set of chromosome, the DNA content increases by a proportional factor, and the mutagenic effect of UV light peaks at 253.7 nanometer, the peak for the absorption of UV light by the DNA. These are the circumstantial evidences which suggest that DNA is the genetic material, and we should also recall from our knowledge beginning from that of Mendelian genetics to the chromosomal theory to the cell division and to the chromosome structure and function, that it is the DNA which is found in case of chromosome, and therefore DNA is the genetic material. Nucleic acid structure is basically, the nucleic acids are polynucleotides. Poly means many. A nucleotide is a combination of phosphorus, sugar and nitrogenous bases. Their building blocks are nucleotides. The nucleotide structure is shown here, that it is phosphate, it is a ribose or deoxyribose sugar, and the nitrogenous base, which are of two kinds, the purines and the pyrimidines. The purines are of adenine or guanine type, and pyrimidines are cytosine, thymine and uracil, and they are popularly referred and identified by their first alphabet, adenine as A, guanine as G, cytosine as C, thymine as T and uracil as U. And the combination of phosphate, sugar and these one of the nitrogenous base is called a nucleotide. And when we remove phosphate, the remaining moiety of sugar and nitrogenous base is called a nucleoside. Ribose is a pentose sugar as shown here in this diagram, along with the numbering of the carbon 1 to 5, and the sugar phosphate backbone is shown here. The phosphate group is by P and the sugar is shown by the pentose ring structure. The nucleotides are all oriented in the same direction. The phosphate group joins the third carbon of one sugar to the fifth carbon of the next in line. This position, known as 3 to 5, and determines the direction or the polarity of the chain, especially in case of DNA. Adding in the base, the bases are attached to the first carbon and their order is important. It determines the genetic information of the molecule. This is a complex structure chemical formula of the DNA helix. As we know that Watson and Crick found out and discovered the structure of a DNA molecule as a double helix, and there we found that two chains entwine over each other. Both the chains are made up of the phosphate sugar chains, and along with their nucleotide they are joined together with AT and CG with different kind of hydrogen bonds. What is important here in this case of DNA structure which was discovered by Watson and Crick, that DNA is a double helix, and at the time of the DNA duplication, or which we call DNA replication, the chain opens up and there is a synthesis of new chain on the old chain, and the enzyme responsible for the synthesis of the new chain over the old parent chain is referred as DNA polymerase. The DNA polymerase can only add nucleotide to the 3' end of the DNA, and this causes the new strand to be built in a 5:3 direction. Here this is shown how DNA is replicated with the help of DNA polymerase enzyme, which is shown here in the case in blue color, and the direction is from 5:3, and the new chain is being synthesized over the old chain in the direction of the replication as shown in the diagram. Now we should remember that how carbons are numbered, and on the basis of these numbering of the carbon there are joining of the phosphate group on the sugar deoxyribose or ribose sugar, and nitrogenous bases which are A, G, C or T or U in case of the RNA, that is uracil. And we should also remember the strands are antiparallel. The one strand, as we can see here, is running from 5:3 direction. The other strand is running from 3:5 direction. They are antiparallel, and we can also see that guanine and cytosine are linked with the 3 bond, whereas thymine and adenosine are linked with 2 bonds. So the bonding is stronger between G and C in comparison to thymine and adenine. Synthesis of new DNA strand take place, as we understood earlier, that the leading strand is synthesized as a single strand from the point of origin toward the opening replication fork. The enzyme is again DNA polymerase, and the direction is from 5:3. The synthesis of the new DNA strand takes place that the lagging strand is synthesized discontinuously against overall direction of the replication. This strand is made in many short segments. It is replicated from the replication fork toward the origin. This is a new kind of the DNA replication where different threads or lagging threads of DNA which appear during the cell division or during the cell metabolism, they are again joined together with the help of certain enzymes, especially the DNA polymerase and the other enzyme known as ligases, and these fragments are called Okazaki fragment, because the Japanese scientist Okazaki discovered these separate fragment of the DNA and their mechanism of joining together. The enzyme ligase joins the Okazaki fragment together to make one strand. So here we summarize that the DNA molecule is a double stranded molecule, and during its duplication and replication the chain opens up, and on the old chain the new chains are synthesized. Because there is always a combination of old chain with that of new chain, this is called as semi-conservative. Semi-conservative means a combination of old and modern. Idea presented by Watson and Crick that the 2 strands of the parental molecule separate and each act as a template for a new complementary strand. New DNA consists of 1 parental original and 1 new strand of DNA as shown here in the diagram, hence it is referred as semi-conservative. DNA damage and repair is because of the chemicals and ultraviolet radiation damage the DNA in our body cell. Cells must continuously repair damaged DNA. Excision repair occurs when any of over 50 repair enzymes remove damaged part of DNA, and DNA polymerase and DNA ligase replace and bond the new nucleotide together. Now at this juncture what is important to understand, that there is continuously a process going on in the cell, that is of DNA damage and repair. So many things damage the DNA and breaks into fragment which we call Okazaki fragment, but there are inbuilt mechanisms for the repair of these damaged DNA, and the enzymes mainly responsible for the repair and joining back of the DNA segment is the DNA polymerase as well as the ligase enzyme systems. Now we come to the another most important part, that is protein synthesis. Before we start to begin our understanding about protein synthesis, we should realize that there is the gene-protein relationship is the most important thing in biology or in the biochemical genetics. All the genes, they lead to a synthesis of a protein, and ultimately this protein either is a structural protein or it works as an enzyme which leads to the formation of so many other kind of metabolites or the biochemical constants responsible for the cell structure and function. So it is ultimately the protein molecule which is of prime importance in biometabolism or in case of biology, and therefore the control of protein synthesis by DNA or by gene is the basis of our understanding of biochemical genetics or biochemical control of the genes. DNA, as we know now, DNA contains genes, the sequences of nucleotide bases. These genes code for polypeptides, that is proteins, and proteins are used to build cells and do much of the work inside cells, which we have again reiterate that proteins are either the structural protein or the functional protein. Enzymes are the functional proteins, and structural proteins contribute in the growth and development of the cell and the plant body. Genes and proteins are made of amino acids linked together by peptide bond, and there are more than 20 different amino acids that exist. Here the structure of amino acid, that there is amino group, there is a side chain, and there is a carboxylic acid group, and the combination of all these three makes a amino acid. The COOH group is the characteristic acid group of the amino acid, and the amino group is responsible for the name amino acid. By the word polypeptide, we actually mean the protein. Polypeptide are synonymous to proteins. In fact, proteins have many polypeptide chain, and the peptide chain means a single chain of various amino acids joined together. DNA begins the process. That DNA is found inside the nucleus. Proteins, however, are made in the cytoplasm of cell by organelles called ribosomes, and ribosomes may be free in the cytosol or attached to the surface of the rough endoplasmic reticulum. Now, starting with the DNA's code must be copied and taken to the cytosol. In the cytoplasm, this code must be read, so amino acid can be assembled to make polypeptide, that is protein, and this process is called protein synthesis. RNA has got a very important role in protein synthesis. That DNA is the master plan and RNA is the blueprint of this master plan. This blueprint is coded, and this process is known as translation, and RNA differs from the DNA in the sense that RNA has a sugar ribose, whereas DNA has a sugar deoxyribose as shown in this figure. And the other difference is that RNA contains the base uracil and DNA has thymine. So instead of T, the RNA will have alphabet U, and RNA molecule is single stranded and DNA is a double stranded, as we all know. Three types of RNA are there: the messenger RNA, which copies DNA code and carries the genetic information to the ribosome, then ribosomal RNA or rRNA, and the transfer RNA or tRNA. They, along with protein, makes up the ribosomes. The transfer RNA transfers amino acid to the ribosome where proteins are synthesized. Now we shall see that how these different kind of RNA help in protein synthesis. The messenger RNA is the long straight chain of nucleotide, It is made in nucleus. It copies DNA and leaves through the nuclear pore, and it contains the nitrogen bases as A, G, C and U instead of T, that is adenine, guanine, cytosine and uracil in place of thymine. The messenger RNA or mRNA carries the information for a specific protein. It is made up of 500 to 1000 nucleotide long sequence of 3 bases called codon. AUG codes for the methionine, which is a start codon. UAA, UAG or UGA are stop codons. Now we are coming toward the crux of protein synthesis, that is the sequence of 3 nucleotide bases is known as a codon. The entire sequence is called code, and this code is transferred to messenger RNA and then used in protein synthesis over ribosomes. The ribosomal RNA is a single strand, that is 100 to 3000 nucleotide. It is globular in shape, made inside the nucleus of a cell. It is associated with protein to form ribosomes, and this is the site of the protein synthesis. The genetic code can be defined. A codon designates an amino acid. An amino acid may have more than one codon. There are 20 amino acids but 64 possible codons. Some codons tell the ribosome to stop translating. Now again we should revisit our knowledge and understanding that the sequence of nucleotide on DNA, the sequence of nucleotide on messenger RNA, sequence of nucleotide on the head of the transfer RNA is the code anticodon reaction, and because amino acids are linked to the transfer RNA, the sequence of the amino acid is determined by codon, and that is basically the genetics of the protein synthesis. And this is a diagram which shows in short the entire possible combination of the genetic code. And now we can see how to form a genetic code. There are 4 in the center, that is G, U, A and C, and there are many in the second round and there are many in the third round. And by making a combination of anything like say G, C, U, and it will form the adenine, the amino acid. In the last lightly colored circle there are names of all the 20 amino acid, and therefore if we use the code by reading from the center to the outside, we will reach to the type of amino acid which will be used in the protein synthesis. For example, AUG codes for methionine, we can see in this diagram. The complementary bases are on DNA, they are A to T and C and G, and on RNA as we know the thymine is replaced by uracil, it is AU and CG. The transfer RNA tRNA is a clover leaf shape. It is single stranded molecule with attachment site at one end for an amino acid, and opposite end has 3 nucleotide base called the anticodon, which is shown here in this diagram. Here at the tail their amino acids are attached to the site of transfer RNA, and here it is the anticodon, the UAC, and we can see that how the codon anticodon reaction occurs and how the amino acid will be placed on a particular site, that the 3 bases of an anticodon are complementary to 3 bases of codon. The example of codon is ACUE, anticodon in UGA, and here it is a particular amino acid caused by UGA. The transcription and translation are the processes of the protein synthesis, and this is the pathway to making a protein, DNA to messenger RNA, messenger to transfer RNA to protein. In protein synthesis, the production or synthesis of polypeptide chain is protein. It is in 2 phases, transcription and translation. Messenger RNA must be processed before it leaves the nucleus of the eukaryotic cell. Like here, the DNA codes for RNA and then to protein. One process is called transcription, another process is called translation, and in case of eukaryotic cell, we see that after transcription, RNA processing and translation, proteins are synthesized with continuous elongation of the amino acid chain. The transcription is the process of copying the sequence of one strand of DNA, the template strand. The messenger RNA copies the template and requires the enzyme RNA polymerase. This is again the description of transcription and the result of transcription as introns and exons, and the diagram shows that how the messenger RNA is synthesized from one template of the DNA, and this is how the messenger RNA comes out of the nucleus from the nuclear pores after the process of the transcription. The translation is the process of decoding the mRNA into a polypeptide chain. Ribosomes read messenger RNA 3 bases or 1 codon at a time and construct the proteins. This is how the DNA double helix is transcribing over messenger RNA. Messenger RNA is getting translated, and with the help of the transfer RNA we see different kind of amino acids are joining together. Here in this diagram, arginine, tyrosine and tryptophan are in the sequence or the result of codon-anticodon reaction of codon 1, codon 2 and codon 3. Ribosomes are made up of a large and a small subunit composed of ribosomal RNA 40% and protein 60%, have two sites for transfer RNA attachment, and here we see in this diagram the two subunit and the joining of messenger RNA transcript. The step 2 is the elongation of the chain. As ribosome moves, two transfer RNA with their amino acid move into site A and P of the ribosome. Peptide bond join the amino acid. So in this way, the ribosome move on the messenger RNA template. There is a continuous codon-anticodon reaction. Along with that codon-anticodon reaction, there is a growth in the chain of the amino acid, which is the growth and the building of a polypeptide chain. And here is the end product, that is the protein. The end product of protein synthesis is primarily a structure of a protein, a sequence of amino acid bonded together by peptide bond. And here we see that different kind of amino acids are joined together to form a polypeptide chain, and when these polypeptide chains rearrange themselves in primary, secondary, tertiary and quaternary structure, a complete protein molecule is formed, which will either be a functional protein or a structural protein. In today's part, we learned the biochemical basis of the genetics, and in that case we learned that it is the DNA which forms the chromosome, and DNA is basically the gene, and it is the DNA which transcribed into messenger RNA and ribosomal RNA and transfer RNA, and by a process which we call as the biochemistry of the protein synthesis, the DNA molecule lead to the synthesis of different kind of protein. The different kind of protein are basically the different sequences of amino acid in a polypeptide chain, and here in this part we learned that how DNA sequence is translated into messenger RNA and then into transfer RNA, which leads to the formation of the various chains of the amino acid, that is determined by basically the sequence which we known as code and anticode on the transfer RNA. Code-anticodon reaction determines the sequence of amino acid, and ultimately the protein molecules are synthesized from the DNA.